This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 1105 or 1105, take your choice, recorded on April 12, 2024. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody. Here it's 50, partly cloudy, and windy, very windy. We had a wind warning, and 50 Fahrenheit is approximately 10 degrees Celsius. And it shows rain on the Norwegian Your Rain weather app, but it's not raining.
So my thermometer says 16C, and it says Chelsea. You know, this is 29th Street, so I think that's Chelsea. Also joining us from Western Massachusetts, Alan Dove.
Good to be here. It's 61 Fahrenheit, which is 16C. That's one of the easy temperatures to remember. And overcast, and the wind blew through here like a freight train last night, but it's been a little bit calmer today.
And Alan, you disputed that this was Chelsea the other night.
I thought Chelsea was below 20th, but
no,
maybe...
it's not. If you look at the map, it's 29th, I think, is the border.
Okay.
Also joining us from Austin, Texas, Rich Condit.
Hi, everybody. 79 degrees. Cloudless sky, light breeze. It's gorgeous. Just spectacular.
And from Madison, New Jersey, Brianne Barker.
Hi, great to be here. I'm also on team 61 Celsius, 61 Fahrenheit, 16 Celsius. And it is gray and cloudy and yucky outside.
I didn't know if it was cloudy. Yeah, it is cloudy. Windy. It says windy with pictures of clouds. Huh. All right. First of all, if you enjoy these programs, there's a noisy cat. If you enjoy these programs with all kinds of cats, please consider supporting us. We'd love your financial support. You can go to microbe.tv/contribute for more information about that. Well, viruses of microbes. I think I talked about that enough, didn't I? Anyway, I don't have any information here. It's all gone. But that's fine. So, Kathy, what do you have about ASV?
Well, I want to remind people that ASV is hosting a webinar this Tuesday. So depending on when you're listening to this, it might have already happened. It's a free webinar, and we will be posting the recording later on our website. But it's going to be hosted by an immigration lawyer, and he's going to be able to tell you about visas to come to ASV 2024, visas to go to ASV 2025, which is in Montreal, and also answer questions, and that could be about other kinds of visas as well. So it's free podcast. You just need to register. So you can go to ASV homepage and scroll down to a section called The Latest from ASV and look for Navigating the U.S. Immigration System. And then it's possible that Vincent will put a link in the show notes to this as well.
Yeah, I will, for sure.
Okay. And on Tuesday, this webinar is 1 to 2 p.m. Eastern Time on Zoom. Then the other announcement I want to make is about upcoming deadlines. Oh, my goodness. I mean, it's already halfway through April. Your taxes are due Monday, and early bird registration for ASV 2024 ends on April 22nd. And then before that, next Friday, a week from today as we're recording, on April 19th, if you're an ASV member and you're interested in dependent care, you can apply to ASV Cares. And or if you're interested in child care on site at the meeting itself, you register with Kitty Corp. And those deadlines are both April 19, and you go to the ASV.org meeting site for ASV 2024, and then click on one of the drop downs about childcare and things, and you'll find how to do that. So deadline, this webinar is Tuesday, and then Friday is the childcare and Kitty Corp and ASV Cares application, and then the following Monday is early bird registration. And registration goes up a big chunk of money, so you don't want to delay. You don't want to miss that deadline.
Okay, we had this morning an email from Giuditta Di Lorenzo, who's a research fellow and loves TWiV and is working in Italy at the National Research Institute Area Science Park and organizing the International Conference, Pandemic Preparedness, Achievements, Current Challenges, and New Frontiers from 11th to 13th November in 2024 in Trieste. Ooh, cool.
Nice.
That could be fun.
Yes.
I know. When I looked at this, I was like, ooh.
All right.
Trieste
all
is a
right.
great meeting venue.
Really?
Oh, yes.
Never been.
Oh, yeah. Multiple times.
Giuditta, why don't you have a TWiV there? Anyway, this is part of the pathogen readiness platform of things with all kinds of acronyms that you don't need to know about. But the purpose is to provide an overview of known and emerging viruses, their potential epidemic risks, foster knowledge, sharing and networking, encourage the development of broad synergies, and multidisciplinary cooperation, which will all be forgotten once the pandemic begins. No, that was my addition. Sorry. We will talk about diagnostics, epidemiology, surveillance, drug discovery, preventive approaches, with the aim of implementing synergistic, innovative, and effective response strategies to tackle the challenges of reemerging infectious diseases and respond to future pandemics. So there's a website for the event. There's a social media post on X. We'll link to that. And a list of invited speakers. Let's see, who do I recognize here? Yoshikawa Oka, Marion Koopmans, Johan Nates, Rino Rapuoli, Emma Thompson. I recognize them. And there are others too.
I would be pretty excited to hear from Tulio de Oliveira.
Mm-hmm.
Julio de Oliveira. Okay. That's from South Africa, right?
Mm-hmm.
Yeah. Oh, right. I know that name. So if you're interested, there'll be a link in the show notes for that meeting. It sounds really good. I think you should have a TWiV there.
Yeah, they haven't asked us to put this on the ASV website, but or at least I don't see it there. So I can see if we can add that. Okay.
And don't forget Dixon's book, The New City, at despommier.com. And I think it's Kathy who found a blog post about it.
Actually, it was Dixon who sent it to us.
He sent an email out about the blog post.
Yeah.
But Vincent doesn't read his emails, so that's okay.
Well, I do. I read today. He wrote, I'm not going to be on TWiV, so I read that.
Okay.
Yeah. Otherwise, we'd be still sitting waiting for him. Yes, there's a blog post which you can check out. We'll link to that. Okay, in the news, courtesy of Amy Rosenfeld.
Hang on. Just
yes.
one comment out of curiosity. I looked up this name. Giuditta, or I don't know how to pronounce it.
Giuditta. Yeah.
It means from Judea.
Oh,
Its origin is Hebrew, and it's a girl's name.
Mm-hmm.
It's like Judith, kind of.
Mm-hmm.
Yeah. Well, you never know, Rich.
No.
You never know.
Well, I looked at this and I said, Guiditta Di Lorenzo. And I'm thinking Italian or whatever, so I want to say Guidita. Right? But
no, Giuditta. Giu is Jew. Giuditta.
Okay. There you go.
But yeah, it's typically a woman's name, but I could be, Devin can be a man or a woman. Who knows? It could be, both men and women have that name. So anyway, in the news, courtesy of Amy Rosenfeld, we got a couple of reports of avian influenza in dairy cows in other states. Remember, we had Texas originally. And now we have North Carolina. That's South Dakota. North Carolina.
Also Michigan.
No.
Yeah, Michigan also. Many states are going to have this. So North Carolina, in a North Carolina dairy herd. This is the National Veterinary Services Laboratory. HPAI has previously been detected in area herds in Texas, Kansas, Michigan, Idaho, New Mexico, and Ohio. Wow. Movement of cattle from affected herds in these states to North Carolina has been suspended. Yeah. And then we have North Dakota. Highly pathogenic avian influenza detected in South Dakota. South Dakota. A dairy herd. Okay, that's interesting. But perhaps even more interesting is H5N1 pathogenic virus, influenza virus throughout New York City. This is a bioarchive preprint. Detection of clade 2.3.4.4b. Like they're playing a game with SARS-CoV-2 variants to see how many dots they can have.
It seems like an IP address to me.
Yeah.
Like to connect to your router.
Anyway, several people who are recognizable here. We have Florian Kramer and Christine Marizzi. Highly path flu arrived in North America in the winter of 2021-22. They spread across the Americas. Here we conducted surveillance of avian species in the urban environment in New York City. We detected highly pathogenic H5N1 viruses in 6 samples from 4 different birds, performed full genome sequencing, shows the presence of multiple genotypes. Our work highlights that the interface between animals and humans that may give rise to zoonotic infections or even pandemics is not limited to rural environments. I didn't know that Wuhan was a rural environment.
No.
Right? It can happen anywhere, sure. Extends into the heart of
Four
our
Corners
urban
virus,
centers.
it was called, and many.
So that's quite interesting, yes. And next, here we have SIDREP. Quick takes, U.S. measles cases, top 100. No, polio, she wanted to highlight. WHO extends polio emergency. Statement today, WHO said some of the factors involved in extending the health emergency, polio, include wild poliovirus type 1 infections in critical areas in Pakistan and Afghanistan, with lineages seen in Afghanistan in 2022 now being detected in Pakistan in 23 and 24. Also, the agency said the risk of spread of circulating vaccine-derived type 2 remains high, with ongoing cross-border detections in Africa and a new emergence in South Sudan, despite never having used the type 2 vaccine. Vaccination gaps are still a threat, given weak immunization and lack of access in northern Yemen and Somalia, which have large unvaccinated populations. Okay. Then we have ChatGPT may help combat vaccine hesitancy. A new pair of pilot studies suggest ChatGPT could be an effective tool to fight vaccine hesitancy. It's a study being done by physicians in Singapore. They asked the software to answer 15 commonly asked questions on vaccine hesitancy, also about sexually transmitted infections. And the answers were assessed for accuracy. And so it was accurate, they say, but there were some problems, and so they want to fix it.
I guess I'm just sort of surprised that the assumption is that people are going onto ChatGPT and asking it things they don't know and having an answer, like instead of Google.
I think that may be a thing. If they Google something and they don't get an answer that they understand or that they like, they might go to ChatGPT. It's kind of like a magic 8-ball.
Okay.
Yeah.
So, how does ChatGPT discriminate between the truth and disinformation?
That is a darn good question, Rich.
Because my immediate reaction to this was you could just as well, as a matter of fact, if you were, inclined, you might even be able to somehow tilt the universe so that ChatGPT amplified the disinformation, right?
Yes.
Man, yeah.
Yes. I don't trust this at all. So one issue is the way you ask the question could have an influence. And also, ChatGPT is just mining all the text it can find from whatever database and online, and if that information starts to get skewed, then I would fully expect that ChatGPT would reflect the skew. It is it is not an intelligent entity. It is just a text reprocessor.
It's interesting to know that it gives correct information right now,
That
at
it
least.
currently gives correct information. That's
I
nice.
don't know that I would have predicted that it would be giving correct information, but it does,
Right.
so that's cool.
Yeah, that's good.
So at TWiV, we have this thing where we have people solve clinical cases, right? So I always ask ChatGPT what they think. And we had one last week, which was someone had acquired diarrhea from a trip in Florida. And so I gave the symptoms, and it came back with Giardia. But then when I added swimming pool, it had a totally different parasite. It had Cryptosporidium. Cryptosporidium. So just the addition of the swimming pool changed the outcome. It was really interesting. So you have to be really careful with your query.
What was the correct answer?
Cryptosporidium.
Okay.
Because, usually it's very funny. So one of the symptoms was the stools were not floating, nor were they stinky. And Giardia makes floating, stinky stool, apparently. The first answer, ChatGPT, even though people say Giardia does this, sometimes it's not stinky and it's not floating. But the pool was the thing, because you will not get Giardia from a pool. But you can get Cryptosporidium from, disgustingly, other people who have diarrhea going swimming.
Yep.
Humanity, what's with you? Okay, then we have viral hepatitis report worldwide from WHO. Did you know that viral hepatitis claims 3,500 lives globally every day? That's a lot.
That's a lot.
And a large proportion of it is Hep B.
Mm-hmm.
Huge, Hep B.
Which is preventable.
Let's
Got a
see.
vaccine.
So the cases went, there's 187 countries. The deaths from viral hepatitis went from 1.1 million in 2019 to 1.3 million in 2022. And as Alan says, 83% were caused by Hep B, 17% by Hep C.
Yeah, I was in a sort of webinar or sort of panel discussion that included some scientists in some developing countries. And they were talking a lot about education about Hep B and details about Hep B. And I was kind of like, is it really that important? And because I had forgotten sort of how much of a problem it was, in the absence of vaccine or in some other places. It wasn't one, it's so rarely one that I think about. But it was made very clear to me very quickly in that panel discussion that was a luxury I have in the US.
Yes, it is.
All right, then we have Puerto Rico declares a public health emergency as dengue cases rise. So
again,
500. Sorry, go ahead.
I said again. This is not their first time around with this.
549 cases so far this year. Half of them in San Juan. Historical records. So it has no impact on travel, which is good because, well, it's mosquitoes that are transmitting it, right?
Right.
So that's not good. And, I don't know if they vaccinate very much there. Does anyone know?
Don't.
I have no idea. They don't?
It works out. It turns out that this semester in my virology class, I have a student who is from Puerto Rico. And he was able to give us a firsthand account of dengue when we were learning about flaviviruses. And so I learned a lot of new things about dengue this year.
Dengue is really bad in Brazil right now, too. Louisa in my lab told me that you can't get insect repellent. It's like until May. It's back ordered.
Wow.
So, yeah.
Then we have an item apparently especially for me.
Yes. So this came through my news feed. The Potomac River in Washington, D.C. is rising higher and higher with the high tide, and this is partly sea level rise from climate change, and it's partly subsidence of the land, which is happening all around the Chesapeake Bay. But the upshot of it for this story is that some of the cherry trees that Vincent loves so much, that are particularly close to the Potomac River, are now being inundated and are dying off, and they're going to have to remove those trees.
For those of you who may be relatively new to TWiV, this is an ancient TWiV arc.
Yes. Vincent doesn't like cherry
that is
trees.
Vincent dissing the cherry trees.
Yes.
I don't want to see cherry trees dying, but they have
No,
to cut
no.
them down.
Right.
158 trees. But, we were talking a few years ago about how people flock to D.C. in the spring to see these cherry blossoms, and I just think they're gaudy.
Those things originally came from Japan, right?
Yes.
As a gift. And
Yes.
in his recent interaction with the Japanese, is he president or prime minister?
Prime Minister.
Prime Minister. The U.S. has been gifted another 150 cherry trees to replace those lost. You'll be happy to hear that, Vincent.
Nice.
Maybe you
I
could
didn't
go
hear
down and witness their planting or something.
Oh, yes. Yeah, I'll go and we'll film it for TWiV, yeah. No, the sad part is that the water is rising. It's
Yes.
not good, right?
Well, I read a little about this. Yes, the water is rising, but 80% of the problem is the basin sinking.
Yes.
The
I
basin
see.
has sunk like 5 feet, and something like 5 feet, and the water has risen a foot.
Yeah, and this is something that's going on all around the Chesapeake Bay. They're actually going to lose a couple of islands that are inhabited, and people who own waterfront property. Nice problem to have, but at the same time, they're having the houses that are much of their money inundated because the land is sinking. And it is, that's 80% of the problem. The other 20% is that the water is getting higher.
Yep. And another one from Alan.
Yes, I thought this was interesting. Sort of a coda to a fascinating life. A fellow named Paul Alexander, who was the last surviving person to still use an iron lung. He got infected with polio in 1952. And there are various technologies now available, obviously, in terms of respirators, but he had found that the iron lung was the best for sleeping, because you don't have to have a tube down your throat. And so he had been spending a lot of his time in an iron lung since the age of 6.
Yep.
Went to law school, had a career as an attorney, lived a full life, and was... had to spend a good portion of his day in an iron lung the whole time. But he has now passed away at age 78.
Yeah, I read some of a story about this a little bit ago, and it talked about him as Polio Paul. I had not heard of Polio Paul before this. But one of the things that I thought was really strange is that he had to train himself to breathe for short periods of time outside of the iron lung to do all of these things. And in one of the stories, they were talking about how he would have blocks of time where he would be outside of the iron lung to do different things, and he would get a reward for being outside of the iron lung. And so it was like, if he could be outside of the iron lung for certain amounts of time, he would get a puppy.
Yeah.
And I thought that was sort of a cute idea.
Yeah. Yeah. He trained himself to force air down with his mouth and throat so that he could kind of inhale that way, and then he could exhale. But he could only do that, obviously, by paying close attention and deliberately focusing on it, whereas most of us just walk around and breathe without having to think about it.
Interestingly, on this article, there's, they have other articles that you should look at. There's one here. The dream of wiping out polio might need a rethink.
Yeah. Yes.
You think?
Yes.
Alexander says, I'm crippled in most people's minds except mine. I'm Paul Alexander, human being.
Yeah.
It's good.
It's good.
It's good.
I like that very much. Human being. All right. That does it for our In the News. And now, speaking of hepatitis, we have a hepatitis paper. Hepatitis B virus. Origin from Nature Communications, origin and dispersal history of hepatitis B virus in Eastern Eurasia, from a group out of a variety of places in China and Mongolia and Russia. I think I got those all.
And there's
the first
also someone from the Max Planck in
Max
Germany.
Planck. And the first author is Bing Sun, and the last author, there are probably multiple
Two
last
first authors, Bing Sun and Aida Andrades Valtuena.
And the last three are Johannes Krauss, Alexander Herbig, and Shinki Kui. So this is hepatitis B virus. So there are two people here who work on DNA viruses. Do you want to do the background, since I'm an imposter?
Ooh.
Either Rich or Kathy. If
Kathy?
you don't watch.
Yeah, I don't teach hepatitis B,
Okay.
so I'm, yeah.
You know.
Okay.
Wow. It's been a while.
Well, I do teach it, so it's okay. I'll correct you.
Small, partially double-stranded DNA virus. Okay, so in the capsid, it is kind of a double-stranded DNA virus, except one of the strands is not complete. Small, what's about 5 kb?
So.
Naked capsid virus.
No, it's got an envelope.
It's got an envelope. Oh yes, of
Yeah.
course. It's got the envelope with the S protein in it, the surface protein in it. It is described by those in the know as a retrovirus with a bad sense of timing. Okay, because it replicates by alternating RNA and DNA as informational molecules. So it gets into the infect cells, is transcribed into an RNA molecule, which
first it's repaired.
First it's repaired, right,
into
by
fully double-stranded
cellular
DNA.
machinery. So it's fully double-stranded. Then I am pulling this out of the archive. And then the RNA molecule can serve as both a messenger RNA and a template for, and one of the encoded proteins, in addition to capsid proteins, surface proteins, is a reverse transcriptase. And the reverse transcriptase can copy the RNA molecule into new partially double-stranded DNA genomes.
When I talk about it in class, the two things I point out that are different than retroviruses are, first, that it doesn't integrate.
Right.
And I kind of say that, retroviruses do reverse transcription as like one of their first steps, and hepatitis B does reverse transcription as one of its last steps. So
Right.
it's exactly what you were saying about having a poor sense of timing, but also doesn't integrate.
So, and it preferentially infects the liver and causes liver disease. And I think one of the most insidious things about it is that it can cause chronic infections, chronic asymptomatic infections even, so that you can be cooking and shedding virus without knowing it, which of course significantly impacts, enhances transmission. And interestingly, a factlet is that the earlier you get infected with the virus, the greater the probability that you will establish a chronic infection. In particular, one of the modes of it, by the way, it's transmitted, depending on the culture, in the U.S., it's transmitted through fluids and blood products, okay? So in the U.S., one of the major modes of transmission is needle sharing and people taking intravenous drugs. It was a problem in the blood supply for a long time before there was a good test for it, and it's now screened against it. But it can also be transmitted perinatally, okay? So an infected mother can transmit the virus to an infant at birth. And under those circumstances, the probability of establishing a chronic infection is as high as like 25% or something like that. There are a certain percentage of these that are just either a mildly symptomatic or more symptomatic hepatitis that resolves, clears, and you're done, okay? But there's a certain number of chronic infections, and the probability of a chronic infection, in particular an insidious chronic infection, is higher if you're infected at birth. There is a vaccine that's a protein vaccine, so it does not involve inactivated virus. It's administered in, I think, 4 doses, the first of which happens at birth. And this is one of the interesting things, because the anti-vaxxers get after this. Okay, why should, mother's not infected. Why should my child be stuck the minute they're born with this nasty stuff that you're going to stick in them? Well, it turns out that if you try and screen everybody for hepatitis to prevent the transmission to neonates, you don't get everybody, and it's extraordinarily inefficient and ineffective. And that in order to really reduce that burden of neonatal transmission, the best way to do it, and it's without negative consequence, is simply to vaccinate everybody. Okay, so that's routine now in most locations that I know, and there's a couple of subsequent immunizations.
And if you don't get it at birth, you can get it later in life. Those of us who were born sometime before this vaccine existed can still get it.
That's all of us.
That's it, yes. And babies who were born who didn't get it can get it as a subsequent vaccine. But as Rich said, giving it right at birth is the best way to prevent perinatal transmission. I just wanted to mention one other thing. The chronic infection is not harmless to the host. Before the advent of the vaccine, I think it was the leading cause of liver cancer worldwide, if I'm not mistaken. It may still be. I don't know. We just had the news from WHO that a lot of people are still unvaccinated for this and still developing it. So
there have been a lot of studies as to whether some of the gene products might directly contribute to the hepatocellular carcinoma, and I don't think that's fully understood. But my own understanding is that part of the origin of the hepatocellular carcinoma is this years-long insult to the liver. That's a chronic inflammation that ultimately can spin out a hepatocellular carcinoma. Yeah.
One thing here, and probably at other medical schools too, is that you have to either sign that you've gotten vaccinated, or you have to waive that you don't want to get vaccinated after reading through the fact that it can be transmitted through blood products and so forth. And so employee health is really big on trying to get people vaccinated.
Yeah.
And global issue, right? And as we will see, global issue for all of human history.
Yeah.
Yes.
So recapitulating what Alan said, the first sentence of the paper, I just loved it because it uses a word we don't use very much anymore, but it belongs to an ancient family of hepatotropic DNA viruses with origins dating back millions of years and still poses a major health burden to humans nowadays. Nowadays is a perfectly cromulent word that we don't use much.
It is.
So this paper is all about genotypes. So let's talk a little bit about genotypes. There are 10 genotypes of Hep B, A through J, based on genome sequences, right?
As if it wasn't confusing that we had hepatitis A through, what are we up to now? G or
G probably, G, yeah. So these genotypes are different among different parts of the world. So for example, A and D are globally distributed. E through J are in specific regions. B and C are prevalent in Asia, 95% of infections. B is further divided into 2 groups, as if it's not enough, depending on whether recombination with C has occurred, so B with C. Genotype F is South American indigenous populations. Genotype G, primarily the Americas and Europe. And that genotype was previously shown to descend from the ancient Western Eurasian Neolithic to Bronze Age lineage, which is Wenba, W-E-N-B-A. And most of the people who have that are co-infected with HIV.
And the Neolithic to Bronze Age is about, 3,000 years ago, something like that. Bronze Age
Something
is,
like that, yeah.
3,000 to 1,000 BC, something in there.
Genotype I is prevalent in northwestern China, eastern India, Laos, and Vietnam. J was first, this is very interesting, identified in a Japanese patient who was living in Borneo, who had lived in Borneo. And it has the highest sequence similarity with HBVs infecting gibbons and orangutans. So they think a recent transmission event between primates and humans. All right. So the thing that's very interesting about this paper is that the people who transmit are people with chronic infections. So the spread of the virus is linked to human migration. And so it's a good proxy for human interactions. And so we previously did a paper similar to this where they're looking at ancient DNA for various sequences. In that case, it was a hepatitis B. So the first ancient sequence is 2012, which we talked about, I believe. They're talking about 8,000 to 3,500 years ago, the Wenba lineage prevalent in Western Eurasia gave rise to genotypes A and D. And so far, there have been 155 ancient genomes published to date, most of them from Western Eurasia. Okay, Western Eurasia, just think. West is towards California.
So like Europe.
Europe, basically. Europe and a little bit into maybe what's now Russia.
Okay. Only 2 genomes recovered from Eastern Eurasian individuals, 12 from the Americas and 1 from Africa. So there's a sampling bias. And they try and get at that here. They look at 34 complete or partial ancient genomes from modern China, Mongolia, and Russia, going back between 400 and 5,000 years ago. And one of the things they find is that Eastern Eurasia seems to be a potential origin for genotypes B and D. So you know what happens is a virus gets introduced into a population and it begins to diversify. It becomes a different genotype in that because it stays in that population. So that's the founder effect when the virus first goes into that population. So this is a sequencing paper, and I'm going to give you the conclusions. But first, I'll tell you what they did. They didn't do any sequencing themselves. They screened existing data sets, ancient data sets, where they get material from teeth from corpses at different sites. They take the teeth and they drill into them and they pull out some pulp and they get DNA from that. The idea being the tooth is protecting the DNA for many years. And of course, the viral DNA got in there, if you're wondering, because there's a blood supply in the tooth and these viruses are viremic. So they got eventually 34 hepatitis B, 34 hep B genomes. It's from 34 individuals from 17 sites in Eastern Eurasia. Their ages are between 400 and 5,000 years ago. Interestingly, some of these people had mixed infections. 9 individuals had mixed hepatitis B infections, 2 different genotypes. Can give you an idea of the people are all exchanging these viruses multiple times. It's quite interesting.
And this is how you can get recombination events.
That's right. So they do a number of standard things. I'll just go through quickly, then I'll do the conclusion from them. So they do phylogenetic placement. So they make a phylogenetic tree of these new samples in relation to what we have for hepatitis B. And they have a nice map here showing where the 17 different sites are. Now, these are typically archaeological sites. And you can see that on the tree, these new samples are filling in lineages that didn't have a lot of data before. In particular, lineages B and C didn't have a lot, and now we have more in there as well. They calculate the time to the most recent common ancestor. And it depends. The overall, it's like 12 to 15,000 years ago, and it differs slightly for each of the genotypes that they found. They look at recombination to get some evidence, and they can see that certain Bs are recombinants of B and C, and D is a recombinant of A and Wenba, etc. Then they also look at the people. They can tell what kind of person the sample is from by looking at the genome sequence of the person, right? And this is really interesting because they can get a sense of how people were moving around in these different countries and carrying the viruses with them. For example, people with genotype B includes modern Hezhen, Zibo, Mongolia, Tibetan, Japanese, Korean, and Nashi, with the exception of one person who was a nomad. It's just so cool that this one person is not anywhere in particular.
Yeah, and there was what people have learned in recent years is there was a lot more travel and trade going on in the ancient world than was previously appreciated. And it wasn't like these cultures were totally isolated from each other. They were back and forth all over the map.
Right.
So in fact, in this paper, one of the sites is or actually a good bit of the data come from Xinjiang. Is that right?
Yeah.
Which they describe as being on the Proto-Silk Road. So it was a very, very early part of the trade route between Eastern and Western Eurasia.
So it's like a precursor to the Silk Road, right?
Right.
Yes.
Which was not an actual like I-95 kind of highway. It was a series of trade routes that went over land and water and connected these places.
And people walked on them, or they rode horses and that sort of thing.
Horses or camels, or they walked, and they took boats for the waterborne parts. And,
Right. So I'm going to go right to the discussion, and because they actually summarize things here in the data section. They never give you a summary after each section. Some papers do, some don't. They don't here. The next paper, they give you a summary after each data section.
Next paper is really good about that.
Very nice. So just multiple genotypes in two of the studied sites, one in Southeast Mongolia, which was built by Mongol tribes between the 12th and 14th century. And I love when they integrate history. It's just so cool. Yeah. And the second site in Xinjiang, Northwest China, 5 distinct genotypes in these areas, A, B, C, D, and Wenba. And so they say this shows you how much diversity we've had in the past in Eastern Eurasia, which is not the case right now, right? A through D are widely distributed across contemporary Eastern Eurasia, but they say, based on our data, we can see they're already present in East Asia as early as 3,000 years ago. They call it before the present. I never heard that one before. YBP.
Yeah, no, that things BC is no longer a thing, and so it's either YBP or BCE, before Common Era.
Before Common Era.
Okay. And they also find Webna in East Asia, even though genotype G, which descends from it, is rare in Asia and is not detectable in China today. So there's a difference in ancient and modern populations. And the diversity in Eastern Eurasia is much higher than it is now. And what else did they? So they have relationships they see in the phylogenetic tree between their isolates. They say is consistent with the cultural interactions of these societies, including Shanbei, Hun, Sarmatian, Karluk, Golden Horde. And they say the reappearance of genotype D may be attributed to the migration of Shanbei populations and Mongols. So basically, you can see how movement of humans and interactions have really played a big role in HBV.
Be a virus, see the world. Yep.
Mm-hmm.
Absolutely.
There's one HBV factlet that we neglected to discuss, which is that there are effective antiviral treatments for it now too, so that you can actually now cure a chronic infection.
That's right. Let's see. So they're really interested in the human populations, their contacts, and how they underlie these different genotypes. And they say they found ancient genomes of genotype B fall into 2 sublineages, and one 5,000-year-old sequence is basal to all the ancient and modern genotype B. In other words, it's an ancestor of all of those. And they say our human analyses reveal that all people containing genotype B shared a remarkably similar genomic profile, indicating that these people all spread it to different places. And so there's a lot of diversity of genomes 5,000 years ago, and they say maybe that's where genotype B came from. And in comparison, there's only one genotype of A, and there's one sequence from Xinjiang, a 2,895-year-old sequence. That's the deepest branch in the lineage. It could be the ancestor to the lineage A. They also talk about origins of other. So that gives you an idea of how you can use these data, the virus data and the human data, to try and get an idea of how these genomes moved around. They have 3 genotypes in Quanaroo Cemetery, A, B, and D. And one of them is the first genotype A recovered from Eastern Eurasia, and it makes a branch related to Western Eurasian strains. And the others from this site have genotype D. And as Alan mentioned, Xinjiang is on the Proto-Silk Road. And that road witnessed the exchanges of people, cultures, agricultural products, and languages, as well as viruses.
There was a big trade in proto-silk back then.
And viruses, too. The East-West admixture between Northeast Asian and Yamnaya-related populations observed in Xinjiang is more than 2,000 years old. So they think the Proto-Silk Road probably contributed a lot to the spread of hepatitis B virus. And they also talk about the origin of genotype D, which is suggested to be a recombinant between A and Wenba, and they say our sequences provide evidence of geographical overlap of AD and Wenba, so that wouldn't be needed to have a recombinant, right, in order to do that. Genotype 1, I, is a triple recombinant with elements from genotypes A, G, and C, and has only been found in northwestern China, eastern India, Laos, and Vietnam. And today there's no overlap between I and G, so that they did overlap at one time. They recombined and then they disappeared. So they think genotype 1 was introduced during the colonial
Genotype
history
I.
in the modern age. I, right.
It's confusing enough with the letters.
I know. So that's the story here. We fill in more of the Hep B phylogenetic tree. This region, Eastern Eurasia, seems to be the origin of genotypes B and D, and there's a big influence of human migration on dispersal of Hep B in ancient times, which you can get. So you get these DNA samples from teeth, you get the virus, and you get the host, and you can look at the interplay. So that's why I think this is a cool study.
Yeah, and unfortunately, these DNA sources are extremely rare and not evenly distributed across the map, so we can't get the kind of resolution we'd like. But the degree to which you can get this information is really, really amazing.
Yeah.
Thousands and thousands of years
Yeah, I mean,
old.
just going to a 3,000-year-old dead body and extracting the viral DNA and sequencing it, it's amazing.
All right, on to a more contemporary story. This is a Nature Immunology paper where the title is Antibody-Independent Protection Against Heterologous SARS-CoV-2 Challenge Conferred by Prior Infection or Vaccination. So in case you missed it, antibody-independent. That's the first 2 words of the paper. Protection against heterologous SARS-CoV-2 challenge, which you can get from infection or vaccination. So
We don't need antibody.
Well, they probably are important, but
Yeah, they're important.
Yeah.
yes. In young mice, you don't need them.
Yeah. Well, and there's one other piece that at least some other immunologists I know would argue about this, but we'll get
let us know. So this is from a number of groups throughout Italy. We have University of Pennsylvania, University of Iowa, and we have 1 and 2. Valeria Fumagalli. Oh my God, what a great name. Nicole Rava, David Marotta, Pietro Di Lucia are the 4 co-first authors. And you have
Wait,
Stanley
are they? 12 is the authors
12? It's not 1, 2? Oh,
It's
you're
not
right.
1, 2. So I have four co-senior authors
Yeah,
and
right.
miraculously, one first author, I
Wow,
think.
that's amazing. 4 co-senior are Raffaele De Francesco, Marco Bianchi, Luca Guidotti, and Matteo Iannacone. And I picked this paper just so I
Just
could...
so you could say the names, yes.
I
Say
knew
the
that
names.
as soon as I saw it. I said,
Well, I didn't,
"That's
but
fine."
it's lovely. I love to pronounce these things. And also Stanley Perlman is in there. That's and Norbert Party. So, as you know, antibodies have been pushed forward as the thing that prevents, well, they say here, bulwark against reinfection. I would say bulwark against disease, right? That's what we've
Yes.
learned. But they say, antibodies are short-lived and the variants get around antibodies. So do we really need to boost every year because of that? So
And
let's
do we
some...
need to continuously update the vaccine with new strains as
Yes.
they come out?
And
Yes.
should we actually measure something other than antibodies?
And then they say, of course, we know T cells are able to prevent severe disease. And that's been found in many studies. We've talked about them on TWiV. And they can do that in the absence of antibodies. We did a paper not too long ago about that. And the T cell epitopes don't change very much in the variants and so forth. And so they're a great candidate for playing a really important role. He said, but and this is very cool. The experiments to do that, to find the role of T cells, are challenging. Right?
Fraught with challenges.
Fraught
is the
with
phrase they
challenge.
use.
Like they say, if you can make mice that don't have B cells, right? And they don't make antibodies, but they have weird lymphoid tissues, right? The architectures are, because there's no B cells, and so everything's going to be different, right? Right, Brianne?
Yeah. So this is the point that I found funny, and as I was reading this paper, my first thought was, oh my gosh, I have to send this to my sister, because we've had this discussion as she has worked a lot on B cell biology, because she sometimes will yell at me for underestimating B cells, and thinking about B cells as only having the job of making antibodies and doing nothing else. But B cells can do other things, like make cytokines, and B cells do other things. We mostly think about their antibody production, but they are, doing other things besides just producing antibodies.
Yeah.
And so the issue is, if you make mice that are completely deficient in B cells, you got rid of the antibody part, but you've also gotten rid of these other weird mystery B cell functions that my B cell biology colleagues tell me are so important. But so the idea is, how do you separate out the role of the antibody from other roles that B cells potentially might have?
Right. And then there's the other challenge. If you want to do adoptive transfer of T cells from one mouse to another, they say, who knows if they go where they needed to be in the right numbers, right? So all of these are inadequate. So they have another approach, which is very cool. They have a line of mice, DHLMP2A mice. They have B cells. They have normal lymphoid tissue architecture, but they don't have antibodies. There's no cell surface antibodies. There's no cell surface circulating antibodies. So why didn't we know this? When did these exist, Brianne? When did they?
I ended up looking up these mice because when I read the very beginning of the paper, I saw the mice without B cells were coming. And I assumed I knew which kind of mice they were using. And then I saw that it was not the mice I was expecting. It was something else. So I, of course, had to go down a rabbit hole of learning about these other mice. And so I found papers from, 2012 that used these mice. And it was not the first they had used them. It was not the first use of them. There are other times where people are talking about these mice from back to 2004.
Wow. I see. Yeah, they've been around.
Yeah. So the thing that's sort of interesting about these mice, again, they were not what I was expecting when I was looking at this, is that these mice have a genetic change, so they can't make the B cell receptor, which means they have no B cell receptor and no antibodies. But the gene that's put in allows the B cell to get a survival signal. So even though it doesn't make a B cell receptor, it makes it through development
Oh,
because
cool.
it got this survival signal.
Yeah.
So there are B cells, but those B cells don't have B cell receptors and don't make secreted antibodies. I was expecting another kind of mouse that makes secreted antibodies or that makes B cell receptor but no secreted antibodies, and that's not what we have here. These mice are missing both the B cell receptor and the secreted antibody.
So I didn't do the deep dive because I was confident that you would. But so, one of the first things I wrote was, why are they devoid of both of these? So these have been engineered in such a way that they don't make, and as soon as you say B cell receptor, to me, that just means they don't make antibody.
Yeah. So basically there's a locus that is the antibody locus, the sort of VDJ and consonant region, and it can be spliced in two different ways. One to make a secreted antibody and one to make one with a transmembrane domain, which is the B cell receptor.
Okay.
And so the change here changes the DNA so that piece of DNA is never made. And so you both don't have the transmembrane version and don't have the secreted version. But you still have something that can signal to allow the cell to get survival signals and make it through development.
Right, because otherwise, without the B cell receptor, they would be selected out.
Yeah, and so they talk about mice inherently deficient in B cells. That's because they're missing all the parts of the B cell receptor and they don't get a survival signal.
Okay, so they have left something of that whole locus behind that's enough to allow this to happen.
It's actually kind of interesting. They took out basically the D segments of the heavy chain, and in their place, they put in a gene from EBV. And the EBV gene leads to signaling that gives the B cell survival signal, but doesn't allow it to make a full B cell receptor because they've taken out the D region.
Right.
Because the EBV, when it latently infects B cells, it makes them into memory B cells, right?
So basically they've put this ABV gene in place of the D segment. In the heavy chain.
Okay.
So they're like memory B cells, but they don't know what they remember.
Yeah, they have no B cell receptor, no antibody, but they just are B cells that are alive and live for a long time.
So there's more than one copy of D segment, is there not?
Yes.
So do they have to knock them all out?
They do. Fortunately, the D segments are all really close together, and on a pretty short part of the genome. So it's doing really a knockout of kind of one small locus.
Okay.
So basically, they're going to use these mice to show that you can get protection against heterologous challenge. That is a different variant without antibodies, because these mice don't make antibodies.
Right. So these mice can mount a cellular but not a humoral response.
Yeah.
Very cool.
Or in theory, they could do whatever the other B cell functions are.
Right.
Okay.
Although they're not, the B cells can't respond to kind of specific antigen, but the B cells, if the cytokines, for example, that the B cells made were the important part, then the cytokines are still there. It's just not antibodies.
So they used two different kinds of transgenic mice in this study. And the first one is one that's been used by others. It's the transgenic for the human ACE2 gene driven by a cytokeratin 18 promoter. You get a lot of receptor made. And they can cross those onto the DHLMP2A mice. So they have the wild type ACE2A mice and then that crossed onto that background.
So you want to remind people why the ACE2 is important.
ACE2, of course, is the receptor for SARS-CoV-2, and the mouse version does not bind most SARS-CoV-2 isolates. So you introduce the human gene in, and you allow the mice to be infected be susceptible to SARS-CoV-2. And the interesting thing here is that so if you intranasally infect these ACE2 K18 ACE2 transgenic mice, intranasal infection, right? The virus invades the brain, and you get they die of a brain infection, not of a lung infection. But if you aerosolize the virus, if you make an aerosol and put in their cage, it doesn't cause a brain infection. which is very interesting. I didn't know that.
And that apparently they seem to think that's due to the high ACE2 expression in these mice.
So that's part of it, right? So it's a little confusing because, the K18 ACE2 have always been known to die of a brain infection when you intranasally. And it's not a great, but you can use death as an endpoint and then see if vaccination prevents it, right? So it's kind of a surrogate assay. But they're saying if you aerosolize the virus, it doesn't kill them of a brain infection. Although, as you'll see, it does still infect the brain. So they do an experiment where you take these two mice. So there's antibody plus and antibody minus. Let's simplify it, right? So the antibody plus mice. So now you're infecting them with this aerosol approach. They all survive infection without detectable RNA in the brain. So these are the mice that can make antibodies, right? Then if you take the other strain of mice, K18H human ACE2 crossed to that strain that doesn't make antibodies on the B cells, then those are antibody negative. 70% of them die between 9 and 12 days post-infection. And they say they have pronounced RNA in their brain. So they just told me that aerosol wouldn't go in the brain.
It doesn't do it as much, I guess.
Maybe it doesn't kill them of a brain infection, right? But it's still there. But that's the cool thing. So there's some survivors from this experiment. 30% of the mice, of these antibody-negative mice infected by aerosol, survived. Because if you'd used the intranasal, they'd all die. You couldn't do this experiment. So you take the survivors and you challenge them with a different SARS-CoV-2. And then so those are antibody-negative and also positive survivors. And the antibody-negative mice survive. They don't make antibodies against the virus, whereas the control mice, the antibody plus mice, they make a memory response and make plenty of antibodies.
And this is challenged with a heterologous SARS-CoV-2.
Heterologous isolate, yes, different.
So with respect to the brain infection, is ACE2 ordinarily ubiquitous tissue-wise? Is it everywhere? Because my sense was that it was this, as we'll see, because of the engineering that they do down the road, that it was a specific issue with the human ACE2 receptor mice, either the superabundance of human ACE2 in those mice, or, though they don't say this, I was wondering the distribution of ACE2, since it's, I don't know how ubiquitously expressed the keratin receptor is, but at any rate, it's clear that influences the pathogenesis, as we will see.
Yeah. And so they look for infectious virus, they look for nucleoprotein, and they don't find them in these mice. And they can see infiltrates in the lung, immune cell infiltrates in the lung. They can see T and B cells going into the lung after the challenge. They can see interferon being produced, and they can stimulate T cells to make interferon by adding peptides, viral peptides. But there's no antibodies in these antibody-negative mice, yet they completely control infection. There's no infectious virus being produced. It's really, so in the absence of antibodies, the K18 human ACE transgenic mice can pretty much resist an infection with SARS-CoV-2. It's amazing, right? Is anyone amazed by that?
Of course I'm amazed by that.
Yeah.
Although, it does come out to yay T cells.
Yeah.
I've been rooting for the T cells from the get-go.
Yeah.
Okay. And this, this is a really happy result as far as I can tell. It also helps explain, I think, some of the observations that we've had about, that if you boost with the Wuhan strain long after the initial immunization, you get cross protection against the heterologous strains. Okay. Because I think you're just basically boosting this T cell response, which is inherently broad spectrum, right?
Right.
Mm-hmm.
And the big differences between some of the variants are in more antibody epitopes. And so you're not going to see that same variation in the T cell epitopes.
Right. All right. So now they take a diversion from immunology. couple of figures of non-immuno of
Turns into a
molecular
structural
biology.
paper. Yes.
So they say, okay, these ACE2 human ACE K18 ACE are great, but let's make a better model. So they compare the mouse ACE2 and the human ACE2, and at the receptor binding domain, this has been done before, there are 8 amino acid differences, and that some of those at least are preventing the SARS-CoV-2 from binding the mouse ACE2. So they do some modeling to look at that. Then they do some mutagenesis to ask which ones are important, and they find a specific number, and then they construct hybrid receptors with part of mouse and part of human. So basically, the N-terminal part of mouse ACE2 is replaced by the N-terminal part of human ACE2, and that has all 7 human amino acids that are needed for interaction in the RBD, receptor binding domain. They test them in 3T3 cells, and they show that they will bind many of the SARS-CoV-2 variant. Okay, so now they define that this part of ACE2 gene is important.
Yeah.
So then they use CRISPR to introduce the change at the mice, right? Because so far we've done this in cell culture. Use CRISPR. Now they substitute the N-terminus of murine ACE2 with human, and these are now called HY ACE2, human. What is the Y for?
Hybrid,
Hybrid?
Yeah.
I think.
HY ACE2 now, it's a hybrid. And these mice seem to be fine. You can infect them with SARS-CoV-2. You need a little bit more virus. They have reduced virus in the lungs compared to the others, but they have comparable impairment of pulmonary function.
Yeah.
So they think it's a good model, right?
Yeah, I think about this as the previous mice were transgenic. So they basically added in the human ACE2
Right.
gene and a promoter region, but they didn't actually remove the mouse ACE2 gene or do anything to the mouse ACE2 gene. Here, they've actually impacted the endogenous mouse ACE2 locus, which means that it is going to be regulated exactly the way the mouse ACE2 locus is normally regulated. It's going to be in exactly the tissues that mouse ACE2 is normally in. It's just now kind of looking more like the human one. And I also love one of these figures where they're trying to show a model where they're infecting the mice because it looks like the mouse is smelling a really big virus.
I know, it's
Yes.
funny. It's very funny. Little puffs. All right, so now they cross these HY ACE2 mice onto this DHLMP2A mouse that doesn't make antibodies, right? And now they have new antibody minus animals. And now when you aerosol infect them with SARS-CoV-2, they all survive. In contrast to the other ACE2 transgenics they all die, right? Or 70% of them, 70% of them die. And these mice don't make an antibody response. The surviving mice have no antibodies, but they make CD8 positive T cell responses. They peak at about 7 days after infection. And then 30 days later, they challenge them to a different variant. So they first infect them with one, and then they challenge 30 days later with another. And the naive mice, in their control, they have naive mice that have never seen virus before, and they get the virus reproduces in them and so forth. But if they use these, the antibody negative mice, the virus doesn't reproduce in them on challenge in the absence of antibodies. And they can also show that there are T cells there that respond by making interferon gamma when you add viral peptides to them as well. So now you have a model where all the mice survive the first infection, and when you challenge them, none of them get infected, even in the absence of antibody. So here's the little sentence at the end of each section. These data confirm an antibody-independent protective mechanism against heterologous SARS-CoV-2 challenge conferred by a previous infection. So this is mild disease. So they want to know, how about severe disease? Will this also work? So they have a strain, a mouse-adapted strain, MA30, which they say is more virulent in mice. And so they infect their mice with this virus. They see antibodies in the antibody plus mice, but not in the antibody minus mice by 7 days after infection. Both of them show virus-specific interferon gamma-positive CD8 T cells in the blood. They re-challenge them 30 days later. The and the controls are the naive mice. They show body weight loss and stuff. But both primed antibody plus and minus mice survive infection. They remain stable in weight. They have no signs of disease. Their respiratory function is reduced because they are infected now. They have, at most, scant traces of viral RNA. And they can see CD4 and CD8 T cells in the lungs of these mice, both AB plus and minus, and they say they have characteristics of tissue-resident memory cells. So they say using a third independent mouse model, we confirm a mechanism of protection against severe COVID that operates independently of antibodies. Everybody like that?
I
yeah,
love it.
absolutely.
It's pretty cool. And in the next experiment, they say, can we use mRNA vaccines to do the same thing? All right, so they give the mice mRNA vaccines, which, and they use the antibody plus and the antibody minus mice. Of course, the antibody minus mice do not make antibodies, as you would expect. You challenge them to a heterologous virus, B1.1.529, and both mice limit replication in their. So both the antibody plus and minus mice limit viral replication in the lungs, and there are T cells in there, CD8 and CD4 positive T cells coming into the lungs and so forth. So again, mRNA vaccines protect in this model in the absence of antibodies.
And so here you're simulating somebody who's vaccinated but has not gotten an updated one for the particular strain.
That's right.
And
Yeah.
I often think about this. I remember way back when we were wondering about too many eggs in the spike basket, because they're using a spike-based vaccine here. That spike is a very large protein, and thus contains many epitopes for both B cells and T cells. So the one place where I might worry about this in another model is if the antigen was a much smaller protein than spike and didn't have a bunch of good T cell epitopes in addition to the B cell epitopes. But spike is ginormous and has plenty of epitopes for both.
So nevertheless, relative to the infection, we are confining our attention strictly to spike. So
Sure.
whatever we're observing is attributable to epitopes just on spike.
Yeah.
At least in the vaccinated
Yes.
situation.
Right. Which is, in a practical sense, really important.
Mm-hmm.
Yes.
So what's the mechanism of protection in the absence of antibody? So they do some depletions where they deplete mice. So they infect mice, and then 23 days later they deplete them of either CD4 positive T cells or CD8 positive T cells or both, and then they challenge them with a different variant.
Right. Because you could imagine someone saying, well, they still have B cells, even though the B cells don't have a B cell receptor. And so how are the B cells actually responding to a specific virus? I don't know. But maybe it's the fact that they still have the B cells. The B cells are actually what's important, and it's not the T cells. Or maybe it's maybe we can come up with some other cell type. Though again, the other cell types we might come up with are not going to be antigen specific in the same way that adaptive immune cells like B cells and T cells are. But I could imagine people who are anti-T cells who would come at this and say, well, yeah, you've shown that you don't need the antibodies, but that doesn't mean it's definitely the T cells. And so I think it's really important that they have this in here. While, yes, I, as someone who spent a lot of my time thinking about T cells, definitely would go to, of course it's the T cells. I think it's important that they show this for people who were less convinced already.
So
Yes.
there are T cell bigots out there?
Yeah.
You may be exaggerating a little bit, but probably maybe not.
Go play on Twitter.
No, thank you.
Still?
Yeah.
Well, so they find that both CD4 and CD8s are instrumental. Depletion of both really compromises protection. And they say the CD8s are really important for managing either high-dose infection or severe disease, whereas the CD4s are better, are needed for low-dose infection or mild disease. And they say depleting either one does not influence the ability of the other cell type to do its work, right?
So the CD8s are killer cells, right? So they're going to kill virus-infected cells. So their role in this would be obvious to a numbskull like me. The CD4s are helper cells of one sort or another, right? So they're going to be like shaping, regulating the immune response. Is that right?
Yeah.
Yeah, they're making a whole lot of cytokines, and so they are impacting how well other cells are responding. There are stories out there of some CD4s that can do some other things.
Yeah.
But the classic CD4 idea is that it's maybe making interferon gamma, which would otherwise influence the infection.
Right. So they also look at interferon because this is what CD8s do, right? They make interferon gamma.
They do,
So
and one set of CD4s do as well, the Th1 cells.
Okay. And so they treat mice with antibodies to interferon gamma before challenge, and this impairs protection. So basically, they say T cells and interferon gamma are critical for protection against challenge in the absence of antibody. And so those are the experiments. They say they unambiguously indicate that mice lacking cell surface and circulating antibodies are capable of resisting heterologous SARS-CoV-2 challenges post-prior infection or mRNA vaccination. Curtails replication and prevents disease as well. And how interferon gamma works, they say we're not sure. There are a lot of potential things that it could do, and we need to sort that out. But the cool thing is they say we need to re-examine our paradigm of making antibodies, you know?
So
Current vaccine strategies focus on generating neutralizing antibodies, and this may not be the right approach.
I have often thought that the reason that there is so much focus on antibodies is that they're easy to measure. Does that make sense?
Yes, very much so.
Yeah, sure.
And I find that, this is just extraordinarily comforting. I feel more immune than I was before, right? It's really great.
I feel like those things I teach people in class about immunology are true.
Yeah.
I was going to say something. Oh yeah. I also feel like that in a funny kind of way, we got lucky with this one. The vaccine was decades of technology and et cetera, but not all pathogens are as easy to immunize artificially against as this one turned out to be. It turned out that despite our initial reservations, spike alone was good enough. And look at this. It gives both a robust and cross-reactive both B and T cell response. How lucky can you get? It could have been a lot worse. We could still be struggling with this.
I've been thinking that ever since the first results came out from the vaccine clinical trials. I was like, oh my gosh, they all worked. I mean, everything everybody tried using spike, they varied a little bit, but yeah, we got very, very lucky that the pandemic we got at this moment when we had this particular set of technologies was one that was highly susceptible to the approaches we happened to have.
As I said, I don't want to, and none of us want to diss the science, okay?
Oh gosh, no.
Because when people talk about, various cures or things as being luck or miracles, I say, no, wait a minute, okay? There's a lot of really hard work and decades of it and great science here. However, not all pathogens are the same.
No.
And this one happened to be particularly susceptible to the approaches we had at hand.
So I have two questions for the group. So the first one is, these are mice, and as we know, mice lie.
Yeah.
So what do we feel about the applicability to humans?
I feel pretty good about the applicability to humans in that, while there are many places where mice lie, I think the general idea of will you make a T cell response and are T cells able to combat a virus is something that's pretty conserved. When I think about mouse immunology versus human immunology, one of the biggest things that I think about as a difference is in terms of how many past infections the organism has had. And so, if anything, we as humans most of us are not antibody deficient. And so we have both arms, antibodies and T cells that can protect us. I think that the T cells are doing a good job based on these data and other data. But we also have probably other past infections with other viruses that might even further impact our T cell response, which the mice, of course, do not have. And so I would say that this is sort of a good baseline to show that T cells do things. But if I were going to imagine, I would say maybe in humans, the T cells may even be playing other kinds of roles as memory T cells because of other coronavirus infections.
Okay.
I don't know squat, and I would refer to Brianne, but I think these mice are honest. Okay, and as I said before, what I go to is an observation in humans that has always or interested me since the pandemic, which is that homologous boosts the immunity to a heterologous challenge. Now, you can rationalize that in terms of antibodies, but having T cells in there and a whole bunch of T cell epitopes, it makes a lot more sense. I really like that.
I was just going to say that the sentence that Vincent just mentioned about reevaluating and refining the prevailing immunological paradigm was the first one that I highlighted. And then the very next one relates to what you were just saying, Rich. The persistent efficacy of T-cell responses challenges the notion of periodic vaccine updates and regular boosting.
Yeah.
Yeah.
Yep. And that leads to my second question. People over a certain age, maybe 65, 70, they get the latest booster, and then 3 months later they get severe COVID and some of them die. So where are the T cells, or do they not make T cells because they're old?
I think their T cells are less functional.
Okay, because of age, right?
Yeah, I think that they are making somewhat fewer T cells. I don't know if that reduction in number is really the big issue, but the T cells are definitely less functional.
Makes sense.
Yeah, I as I look at this, I also think about this is kind of related to what Rich was saying. This is another point in favor of vaccine types that give you good T cells and good antibodies.
Right.
And so you can think about some vaccine types out there that may be a bit more antibody biased. And this shows us some reasons why the ones that also include T cells might be a good idea.
And because the pandemic is only... sorry, Rich.
Oh.
And Kathy was making a face. I want to know what Kathy's face is.
I'll say something too, but Alan and Rich can go first.
So in that regard, correct me if I'm wrong, but the mRNA vaccines are sort of, with respect to T cell response, sort of self-adjuvanted, right? They do a good job of stimulating a T cell response. And it also puts an emphasis on, with the protein-only vaccines, the adjuvant is really important because that's the component, really, that stimulates the T cell response. If I got all that right?
That is very much right, yes.
Okay, good.
So the thing is that elderly people tend to, we get a lot of emails saying, should I get another booster 3 months later? I don't think that's a good strategy to get boosted multiple times a year. Daniel always says, if you get infected, take Paxlovid, right? And that seems to make sense, but you can't get Paxlovid everywhere in the world, unfortunately. So
there are some that can't take it. But,
some people cannot, right, because of drug interactions. So it seems to me, well, Brian, can you make a vaccine that does a better job at inducing T cells in old people?
I don't know off the top of my head, but I am pretty sure that there are people who are trying to figure that out. And I know certainly if I think about some of the vaccines that are given, they're given at different dosages. in different populations. And so perhaps if older individuals had a higher dose or a different dosing scheme, that might give them more T cells.
So that's what I was going to ask about is, are there people who are actively working on, this aspect of the Holy Grail of, living forever, helping, people who don't want to grow old? Can we do anything to increase T cell function, and not just necessarily against SARS-CoV-2, but against other things? And if you did get boosted, would that keep your T cells like functioning longer because they functioned recently? It's kind of a weird way to ask it, but
I think that those are things that people are working on. I know I've seen some cool talks about some of that stuff, but I don't know that there's a perfect answer yet. But yes, there are definitely people who are thinking about some of those things.
Yeah,
and if nothing else, thinking about, well, can we describe what actually is different and why those T cells are functioning differently so that we could intervene?
Yeah, I'm thinking a simple dietary thing, like could we eat more chocolate or?
I vote for eating more chocolate. Yeah,
sure. Does it? So I was going to point out, the pandemic is this virus has only been in people for about 3 years, and so the people who are in their 70s who are getting vaccinated, they started out with T cells of somebody who was 70-something. And they've persistently not been able to get that T cell response going. If you were younger than that when this came by and you managed to get a robust T cell response, will that also fade? I mean, are the rest of us going to get to be in our 70s and have this same problem, or are we going to be protected maybe a little better because at one time we had a good T cell response?
I think that the answer is that everyone's T cells will sort of undergo some attrition as they age. And so I think that whether you got that first exposure when you were older or whether you got that first exposure when you were younger, as you age, your T cells will not be functioning as well. And one of the things I'm thinking about as I talk about that is thinking about chickenpox and shingles.
Ah, yes.
And if you think about it, people are getting chickenpox and seeing varicella zoster virus pretty young. And they're starting to have shingles when they start to have that attrition of those immune cells, and when the immune cells are no longer sort of able to keep the latent virus in check.
So maybe we need an entirely different vaccine for the elderly population, or a higher dose, as you suggested, that would stimulate that T cell response, regardless of when you initially got exposed, because it's just going to senesce in all of us.
Yeah, I mean, there's already the higher dose influenza vaccines.
Mm-hmm.
Yes.
Right? So,
well, and the original shingles vaccine was simply a double dose of the chickenpox vaccine. So, does the T cell response, T cell immunity, not wane with the same sort of kinetics as antibodies?
There's some debate on that one. My read of the literature is that no, they kind of wane similarly.
Okay.
But I know the paper that I often cite on that is a paper that people like to debate.
Gosh, there's debate in the immunology.
I know, it's shocking.
However, common cold coronaviruses doesn't make anybody die now because they have now infected everyone at a young age, and by the time you get old, you're okay. So it could be that this ends up like that.
Yeah.
Right?
That's why I was wondering if you got infected, vaccinated, etc. at a young age, is that going to have enough momentum to carry you through old age? But we don't know.
It's hard to tell.
We'll see. We'll see in 20 to 30 years
Yes.
when the elders are gone and it's replaced all by younger people who were initially infected as a young person. We'll see if the pathogenesis changes. That would be a population immunity effect.
Right.
Right. And so I was talking to someone last week, and there's this 1889 respiratory outbreak, which everyone has thought was a pandemic. Could have been one of these common cold viruses, coronaviruses, and which now is mild. But back then it was a pandemic. So wait and see.
Yeah. TWiV 10,000 coming up.
Great discussion. This is a great discussion. Sorry, Kathy.
Yeah, I was just going to say, if only somebody could go back and get military fixed formalin samples and try and reconstruct the 1889 virus. That would be
Yes, that's a problem, right? I was
Yeah.
asking, I said, Steve Morse, can we get that? He said, well, there are no samples. Nobody has anything. So
If
But
you have
maybe
samples, please call us.
Yeah.
if you have samples, please call us. Yeah, that's pretty cool. All right. Let's it's 4:30. Let's do some picks of the week. Let's see Brianne is first here.
All right. So earlier this week, I traveled to see totality on the solar eclipse. It was amazing. The travel was not amazing. Is the nicest.
Where did you go?
I went to my parents' house in upstate New York.
Oh, yeah.
So, I mean, they have a big backyard that was exactly in the 100% totality range. It was ideal. I brought a bunch of friends from here. We had a really wonderful time, but it is a 4.5-hour drive. And driving back Monday night with the rest of the people who saw the eclipse, my 4.5-hour drive took 8 hours. So that part wasn't great, but the eclipse was amazing and we loved it. And we were all sort of talking about it as our once in a lifetime experience. And someone asked, well, what about other planets? Is this a once in a lifetime experience? Is this a once in a planet time? Like, would we be able to see this if we lived somewhere else? And so we got very excited about thinking about, is this just us on Earth who get to see these cool things, or could someone else see them? And turns out a few people have tried to answer that question and have actually examined whether other planets have solar eclipses. And so this article kind of goes through some information about which other planets have solar eclipses and what criteria there have to be for that planet to have a solar eclipse. So, of course, first your planet has to have a moon, and then there has to be the right size of the moon compared to the size of your star and things like that. And so it indicates that most planets or many planets have solar eclipses, but not all of them, and makes me feel even more special about having seen that really cool total solar eclipse.
Yeah.
So the other thing that I learned from this is that there's this special thing about the 400-fold, the size difference and the distance that make it work out. So although there might be eclipses on other planets, they might not give you the totality effect that we saw.
Right. Phobos is too small and too close. And
Yeah. And so just the fact that kind of we have the right size moon and the right size sun and they're the right distance apart and all of those kind of things.
And it's a limited time opportunity on Earth. Apparently,
Yes.
in about 600 million years, the moon will have drifted too far away to give you totality.
Oh.
Yeah. So I thought that was really cool to think about. I'd never thought about it before until one of my friends asked if it was special in that way.
Very cool.
I think the word totality is cool.
Yeah.
Is totality cool?
Yes.
Is there a word, totality?
Oh, yeah.
Yeah.
Yeah, for sure.
Oh, yeah.
Yeah, the moment of totality when the eclipse is.
No, but is
the
it
totality of causes of something?
Yeah?
Yeah.
Yeah.
The
The
totality of
totality-
the evidence.
Yeah.
Okay. I only hear it in eclipse context. Kathy, what do you have for us?
Well, first I have to say that I also drove to see totality, but I had very severe time constraints because I needed to be back in Ann Arbor at 7:00. So at first we thought I was overthinking it, but we figured out a place where we could drive down on US 23, get to Toledo, get to almost the northernmost part of where you could see totality. And I looked on Google Maps and I figured out this Home Depot parking lot, and I was worried that it was going to fill up with people because all we had to do was turn right out of the parking lot, turn right out onto the main drag, one traffic light, and then we were on the on-ramp to US 23 and we'd be ahead of everybody else, especially if we left right after totality, which we did. And so we made it back to Ann Arbor, and I was able to see the very tail end from my brother's front yard. So, and we made it to our rehearsal on time. So
Nice.
it was really great.
Yes. Well, if people want to see photos or video, I have.
Yes. So, and there was no one basically in the Home Depot parking lot. So, I mean, there were two other cars that we saw that were there watching, and then there were the employees and the customers who came out of Home Depot and were standing way over there by the doorway. But we were on our path for fast exit.
I wonder what Lowe's was like.
I don't know. Didn't see one of those on the map in the right place. Anyway, so because of it being a week of cool things in the sky, I had seen this one a while ago about this Radcliffe wave, which fascinated me at first just because of the name of it and why was a wave named after Radcliffe. And now am I going to be able to find that quickly? No, I can't find why it was named after the Radcliffe. But so this was a chain of star-forming clouds that washed over the Earth, and it's just a cool picture. Joel Achenbach wrote about it in the Washington Post, and it turns out that this Radcliffe wave is waving. So this was based on a paper in Nature in February. And so our solar system passed through this wave about 13 million years ago, and so that could have led to a festival of supernovae going off. Anyway, so that was really cool. It's got a nice pictures and stuff, and in the link that I put in.
And this structure is 9,000 light years long.
Yeah.
It's a long wave.
Yeah. So this printout that I have doesn't explain why it was called the Radcliffe wave. So I'll find that and get back to you. But then the second one that I picked was this Mount Etna smoke rings.
Yes.
That was, at first I just saw the still picture in the New York Times. And then I figured, I've got to send this to somebody. And then, so I went and found it online, and the video is 40 seconds long. It is awesome. And you just have to click on the video and watch it. It's like this perfectly round thing, and it's puffing smoke rings. It's just amazing. So check it out. So 3 things in the sky this week.
Neat. Well, I didn't see totality. I just went out on the street on 7th Avenue. People were all lined up.
Yeah.
Yeah.
And then I went to campus because I had to teach. And I raised my camera up to take a picture, right? And this guy who was washing the sidewalk, he ran over and said, no, you have to use these glasses. He gave me a pair of glasses I put over the lens, and it worked really well. It was cool. But I like this community feel.
Yeah.
Yes.
Mhm.
Everybody's like watching it. They're all...
We were we saw the total eclipse in 2017 in South Carolina from totality. That was jaw-dropping. But this time, weren't going to do that again. And so we had 93% here, I think, which was actually pretty impressive. I mean, it got dim and the temperature dropped. And of course, I had I had a welder's glass, and Sophie had the glasses from school, the eclipse glasses from school. I had my sextant out, and a couple of people walking by on the street came up, and we showed them. Yeah.
Cool.
I found the Radcliffe wave part. Okay.
Mm-hmm.
They named it in honor of early 20th century female astronomers associated with Radcliffe College, including Radcliffe graduate Henrietta Leavitt, who discovered that the periodic brightening of certain stars encoded information about their distance from Earth. So.
Neat.
Cool.
Rich, what do you have for us?
First of all, we saw totality too. My son flew in with his family from Seattle. But the minute and a half we were going to get in Austin wasn't good enough for him. So we went to Dallas. And we spent, he had it all figured out. We spent 2 nights in a hotel in Dallas so that we wouldn't have to worry about traffic. And watched the eclipse from the Cotton Bowl. And the forecast was miserable, but the clouds parted just in time, and it was glorious.
Mm-hmm.
Cool.
So, my pick.
You found an oak gall.
Look at that. Nice.
Beautiful.
I'm showing, for those just listening, I am showing what Alan properly describes as an oak gall. I had never encountered one of these before. My wife brought it in from out back, and I wondered, okay, what's this? So this actually falls neatly under the category of you can't make this stuff up. And also is, a miracle of being able to just have this computer and the whole internet in your pocket Okay. So what did I do? I took my Seek app and I used that to identify what this is. And it is called a spongy oak apple gall, which is made by the spongy oak apple gall wasp, or Amphibolyps confluenta. And I've given two links here because I had kind of look all over the place to get the complete information. One of my links is to the life cycle, and I just want to read this because this is the life cycle of the wasp. Pardon me, this will take a minute
no
or
worries.
two. The life cycle of this wasp involves alternate generations, one generation with only asexual females and one generation with both sexual males and sexual females. After mating in the fall, the sexual female wasp drops to the ground. She burrows into the soil at the base of the tree and injects her eggs in the tree's roots. These eggs, the first generation embryo stage, overwinter. When they hatch in the early spring, larvae begin feeding on the roots. Soon they enter the pupal stage, a period of inactivity and metamorphosis. Later, still in the spring, the wingless asexual female adult hatches and emerges from the soil. She crawls up the tree trunk, finds a leaf, and injects a single egg into the midrib. Then she finds another leaf and repeats the process with her remaining eggs. These eggs are the second generation embryo stage. The egg hatches and the larvae begins feeding on the leaf. This causes a chemical reaction in the leaf that results in the formation of a ball-like gall. The galls appear in the late spring or early summer. As the season progresses, the larva gets larger and so does the gall. Second generation winged male and female sexual adults emerge in the fall and immediately search out a mate. The cycle continues. Okay, so that's weird enough. Here, now I looked up at my tree and it's got a bunch of these. Here's it cut open.
Oh, neat.
Okay.
Neat.
And there are pictures of this. I'm showing the split open gall, and it's got the larva suspended in the middle with a bunch of fibers projecting out from that, connecting to the outside of the gall. Now, here's the weird part. It turns out, and this is the second link, which I won't read from, but the second link describes some of what's known about the formation of the gall. This is leaf tissue,
It's the
okay?
immune system. Yeah.
And
Yes.
both the wasp and the larva emit chemicals that are thought to be maybe like plant hormones and stuff, and reprogram the leaf tissue to make this gall. Give me a break. It's unbelievable. And interestingly, this gall had apparently just fallen from the tree, and then my wife threw it out in front and went and recovered it today, and it's not green anymore. So, as they say, the gall grows with, during the season. So that's living leaf tissue that has been reprogrammed by both the parent wasp and, I guess, continuously by the larva.
Yep.
Wow.
So why is it called a gall, and what does it have to do with the word gall?
I have no idea.
Alan, do you know?
Well, I don't know if this is the same as the origin. I think the word gall originated with the medical concept of your humors, and if you have a lot of gall, it's because you have too much spleen, I guess, is the I think that may be the origin of this, and this oak gall may be a different etymology. But I do another little factoid about these. Historically, these were used to make ink. In fact, the primary ink in ancient texts all the way up through, into the 20th century, the ink used in regular pens for writing was predominantly what's called oak, iron gall ink. So if you take these oak galls and grind them up, they're very high in tannic acid, and there's gallic acid that's in them. But the tannic acid, if you mix that with iron sulfate, it reacts and will dye paper a dark color when it oxidizes. So you can get iron gall ink. It's actually really cool to write with because it's like a light gray when you write with it. And then as it soaks in and oxidizes with exposure to air, it turns darker and darker blue-black.
Interesting.
So I did some Googling about the etymology here. It is different than the gall, like gallstones and bile, which is from Old English, while the gall that Rich is talking about is from Latin galla, which means oak apple.
Okay. So gall is a word for gall.
Right. There are at least 3 different independent etymologies
Yes.
and meanings of gall.
So
I
gall
Googled
is
it too.
divided into 3 parts.
Exactly.
Yes.
Nice. Show and tell. That's great. Thank you,
That's good.
Rich.
Sure.
And that's why the cat was playing with it. Right.
Yeah, the
Now
cat
I
was
get
split in the beginning of the episode. The cat thinks this is his toy.
I thought
But he's
it was
gone.
like a plushy thing, a virus or something. I don't know. Alan, what do you have for us?
I have, I've linked, there's covered in a couple of news outlets, but I've just linked directly to the DOJ press release on it, which is where all the public information came from. This is an absolutely bonkers story that I thought, wow. This guy in Montana has apparently pleaded guilty to a couple of wildlife crimes for importing parts of endangered species, which is a violation of international and national law. That's not that surprising, but what's so bizarre here is what he was importing and why. So he imported pieces of Marco Polo sheep, which are an endangered sheep species, like a mountain sheep species from Kyrgyzstan, that grows very large and has this grows huge horns, like 5 feet wide. The reason he was importing this stuff was so that he could send it out to laboratories to generate the necessary material to try to clone these sheep, in order to sell them to captive hunting operations that we've talked about in the context of deer. So there are these captive hunting operations where somebody can go and pay money to just walk in and shoot an animal in a pen, and then they can put the trophy on their wall. This guy wanted to clone these giant endangered sheep for use at these hunting ranches. And what's jaw-dropping about this is he got really far in the process. He actually managed to breed a pure genetic male Marco Polo sheep out of cloning from parts that he'd had imported to the country. And he was in the process of trying to cross that and breed out the whole breed that he could then sell to these hunting ranches.
So he actually got a cloned sheep?
Got a cloned sheep, a Marco Polo Argali that he named Montana Mountain King. The guy's in Montana. And then he had he had other unnamed co-conspirators who I gather one of them either screwed up or squealed, and that's how this all came out. And they were going to artificially impregnate other species of ewes and create hybrid animals that could then be sold off to these ranches. And the guy's 80 years old. So I just thought, my God, if this is going on, what else are people getting up to with the availability of this technology? You know, because he's not a molecular biologist.
Who did the cloning for him?
Right, exactly. He's sent this out to some lab somewhere, and that's not specified, but I'm sure that DOJ and FBI and many other agencies are now checking a lot of people's records. So that was a whole scheme, and I'm wondering what we're going to hear of next in this brave new frontier of accessible molecular biology.
Wow. Crazy.
Yeah.
So
Well,
he's I guess 2 felony crimes.
Yep.
Each count is punishable by a fine of up to $250,000 and 5 years in prison.
Right. I suspect since he pled guilty, I think there's probably some kind of a deal being worked out here to identify and deal with the other co-conspirators. And
I think the people who did the cloning ought to.
I think the people in the labs, depending on what they knew and what they should have known, yeah.
This is so funny. Sheep parts.
Yes.
Really cool.
This whole underground economy that you just, yeah.
I have a pick, which I'm sorry to say is from the New York Times, but it's kind of cool. And so I know, it's paywalled. So, but let me give you a sense of what it is. The article is called "These Scientists Rock," literally. The Pasteur Institute in Paris, known for its world-altering scientific research, has been making advancement in another field, the musical arts. This is by Jessica Roy. And apparently some of the scientists have made bands and play music. And they have an on-site studio called the Music Lab. And on a Friday evening in March, they headlined an event at the Institute's cafeteria with names like Polaris and Billy and the Watt, and an a cappella group, Les Papillons. They have some photographs of it here with purple light and 100 people. And sounds like fun.
Nice.
It's spearheaded by a guy, Germano Cecere. 44, was born in a small village near Naples. I bet I know that village because my grandparents were born in small villages near Naples.
What's the herpes virus band called? I keep forgetting.
Herpetic Legion.
Herpetic Legion. Yeah.
And there's a recording of it at an ASV in Wisconsin, I think, years ago. I made a recording. It's on the YouTube channel. You could go see them play. It's very funny. But yeah, that's another example of scientists having fun. So it's cool.
Cool.
One of the group members, Ms. Libri, Alice Billy Libri, is completing a PhD in DNA repair. I just think it's funny to read this article. And all of a sudden, DNA repair comes up. We're talking about music. Music is the main activity. There's a choir. There are guitar lessons and stuff. It's really nice. That's cool.
Yeah, very cool.
Pizzas and a towering cake made of donuts were served. And then Billy says...
Oh, Cesare was postdoc at Columbia.
That's right. He was.
I that's not something I remembered. That's something that's mentioned in the article.
It's in the article.
Kathy sent a gift link to it.
They asked Billy, what's next. She says the music is a way for her to escape when she's disappointed with science. And then she added, I can always go back to science when I'm disappointed with music. It's pretty cool.
Yeah.
I have a couple of listener picks. First is Peter. Oh, the gift link. Thank you very much. Hello from a slightly chilly London. This article about the discovery of artemisinin, an anti-malarial drug in Mao's China, is well written and engaging. It will appear to both the presenters and the listeners of TWiV and is a reminder of the wealth of knowledge hidden in traditional medicines. Asimov Press. I didn't know there was such a thing. Chinese scientists discovered artemisinin in 1971 as part of a secret military project that merged Eastern and Western medicine. That's neat. And it's so it's an article about it, and artemisinin, of course, is a widely used anti-malarial drug. It's pretty cool.
Cool.
They have some old black and white photos. So a lady got didn't she get a Nobel Prize for this?
She did.
Was it for artemisinins or something?
It is. No, it was for artemisinin, and I always mess up her name. I think it is 2UU. I can remember never which one is doubled and which one is singled.
That's right. 2015, 2UU.
Yes.
The same time that Bill Campbell got his, right?
That's why I know a lot about that Nobel Prize from that year.
That's cool. Thank you, Peter. And then Shirley says, this is interesting, and sends a link. This is quite an interesting article. This is in the London Review of Books. A national evil, Jonah Goodman on the curse of the goiter in Switzerland.
Oh.
At the turn of the 20th century, the Swiss were plagued by strange interlinked medical conditions. Endemic in Switzerland, a goiter, and what was the other one? Cretinism. The people, they grow meter tall, had compressed features, thick skin, thick hair, distended bellies. They used to call it cretinism, of all things. Those with the disease were unable to speak or were profoundly brain damaged. So they wanted to figure out what was going on. This is the story of it. It's pretty cool. It's a long story. It's nice. It's nice. It's good science writing, Alan.
Yeah, it is.
And that's it. That's it. Two of them. That's Shirley and Peter. Thank you, Shirley and Peter. If you want to send your pics in, you could send them to TWiV@microbe.tv. And if you like what we do here, again, consider supporting us. We'd love to have your help in growing these science programs. Microbe.tv/contribute. Kathy Spindler is at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun. And I'll do like Daniel does. You can stop the recording right now and make that contribution. And you can stop the recording right now and do your registration for ASV 2024.
I registered, I booked a hotel, and I booked my flight.
All right.
All set to go.
Yeah.
Columbus, Ohio. Brianne Barker's at Drew University. Bio Prof. Barker on Blue Sky. Thank you, Brianne.
Thanks. I learned a lot. And don't worry, Kathy, I have all my registration done too.
Wasn't referring necessarily to the two of you, but everyone else can do it.
Rich Condit is an emeritus professor, University of Florida, Gainesville. He's currently in Austin, Texas. Thank you, Rich.
Sure enough. Always a good time. Great to see you all. Good discussion.
Alan Dove is at turbidplaque.com. Thanks, Alan.
Thanks. It's always a pleasure.
On the stream the other night, Alan, someone asked Alan, all right, what is a turbid plaque?
Yes, and I
Ooh,
explained.
Boy, that's a big question.
It's deep. That's a deep question. What is
Well,
a
I
turbid
put,
plaque?
now that that's my personal blog, I gave it a new tagline. It's a confusing mixture of ongoing projects.
Confusing mixture of ongoing projects. Do you still maintain alandove.com?
I do. AlanDove.com is now my more serious site because I figured that's where people are going to go first anyway.
All right. I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.